Everyone, this is Mrs. Moriarty, and I'm here to discuss with us concept number 4, which is the last concept in our unit of DNA and protein synthesis. This one's going to be all about how we happen to use technology involving DNA. So here, looking at technology used to help with genetic engineering can help us to do a multitude of things, especially since after Watson and Crick have developed the structure of DNA back in the 1950s, we've seen a tremendous boom in how we happen to use DNA and continue to use DNA. So some of the things that we can do with that is maybe identify genes for specific traits, especially ones that maybe happen to cause diseases. We can transfer genes for a specific trait from one organism to another. So this is where we can get organisms called genetically modified organisms. So we can take a desired trait, let's say for strawberries, for example. Maybe we want our strawberries to be larger in size. We can insert a gene or manipulate a gene in the strawberry that would then produce much larger strawberries. We could hopefully identify genes for specific traits in order to cure genetic diseases. We could treat genetic disorders and maybe even improve food crops with each and every one of those pieces of technology. Now, some of the tools used, number one, could be extracting DNA, which we did back when we extracted DNA from a strawberry. You can technically extract DNA from really any living thing. Two, we've developed what's called restriction enzymes. These are enzymes that are specifically built to cut DNA into fragments based on sequences. Three is something called gel electrophoresis, which is a piece of technology often used in what's called DNA fingerprinting, something that can be used to test for paternity tests as well as to solve crimes. And actually what you're seeing in the picture there would be a step-by-step process of how someone might run what's called a gel electrophoresis. Number four is another tool called polymerase chain reaction. This is where we can amplify or make more and more copies of DNA in a lab setting. Now, looking to restriction enzymes first. When we use restriction enzymes, these are technically bacterial enzymes that are going to be used to cut DNA molecules into more manageable pieces. And how they do this is they recognize or they pick up certain sequences of A's, T's, C's, and G's, creating kind of single chain tails in DNA, or known as what's called sticky ends. Sticky ends can readily bind to any complementary chains of DNA. Therefore, our pieces of DNA that have been cut with the same restriction enzyme can basically bind together to form a brand new gene. Here is a picture of that type of process. So here you can cut DNA into fragments, which make those little sticky ends here. And we see that we've now cut two different gene sequences that will eventually be complementary to one another. When that cell goes to then make proteins in protein synthesis, that can wind up producing maybe a slightly different protein. Here would be another picture of that same process, right? Using restriction enzymes, picking up on certain sequences of DNA, creating those sticky ends, and eventually making what's called recombinant DNA. So this is a process that can be used in making genetically modified organisms. Now, another DNA technology technique that we are going to spend some time in class talking about, and as well as modeling, is something called DNA fingerprints. Now, oftentimes, right, we've heard that everybody's fingerprints are slightly different, right? Everybody's DNA is slightly different. So when we say DNA fingerprint, we are talking about patterns or bands of specific fragments of DNA, not necessarily someone's actual fingerprint. What we use DNA fingerprints for are to detect maybe relation with people, right? Like I said, we could use it for paternity tests. We could see if there happens to be similarities between species. So we could use that in investigating evolutionary relationships. And we can also use that to potentially solve crimes. So what are some of the steps involved in coming up with or making a DNA fingerprint? The first thing, DNA needs to be extracted from some sort of specimen, and then eventually a restriction enzyme needs to be used on that DNA. When the restriction enzyme is used on that DNA, that's going to break up the DNA into fragments. And we are then going to use a process called electrophoresis to then separate the DNA amongst its size and its charge. When we do this, we place samples of that cut DNA into wells made up of a gel. And usually that gel is called a agarose gel. And that gel actually has tiny little pores in it. And we take that gel and we actually hook it up to a battery. And we're going to run an electrical current through the gel, hence the name electrophoresis. The negative fragments of DNA are then going to actually be pulled or migrate towards the positive charged end of the gel. The tiny little holes or pores in the gel are going to help to separate those DNA fragments based upon their size. The smaller the fragment, the further the fragment's going to travel through the gel, hence giving us all different patterns and bands when it's all said and done. So what might this look like? Here would be a micropipette that's going to carry our DNA samples. These are the wells. These are little designated areas where the specimen or sample is going to go. And this square thing here would be our gel, and then here would be the electrical currents that we're going to set up when we hook it up to a battery. So here's the negative end where the DNA sample is going to be placed, and here's the positive end, which is where the samples will eventually travel to. Notice how when we eventually turn on the power, DNA fragments are going to migrate through the gel. Fragments that remain closest to the wells or the negative end are larger in size. It's a lot harder for them to migrate. Versus ones that travel furthest, those are technically smaller in size since it's a lot easier for them to travel through the pores. So what you finally get is your DNA fingerprint, right? Patterns or bands of DNA that, as I said before, could potentially be used for a multitude of things, to solve crimes, to look at relationships amongst different species, or even relationships amongst maybe different family members. So in this case, in this DNA sample, looking at what the crime scene DNA pattern, this is probably DNA that was left over at a crime scene from maybe a potential suspect. DNA was gathered and extracted from 3 separate suspects, and a gel electrophoresis was run. So what a scientist here would potentially do would to look for the pattern banding that matches the crime scene. So can you tell who committed the crime from this DNA fingerprint? Correct. It was suspect number 2. We know that it is suspect number 2 because the patterns in the crime scene DNA and suspect number 2 match completely. So that would mean that DNA must have been left behind at the crime scene. Now, the last tool that we need to be familiar with is something called polymerase chain reaction. This can actually be an additional step used in creating a DNA fingerprint, especially when you have a very small sample of DNA. Basically, polymerase chain reaction is used to initiate replication of DNA. So this helps to amplify the amount of DNA that we potentially have. So what can PCR be used for? It could be used for crimes, it could be used for diagnosing genetic disorders from maybe embryonic cells, or maybe even studying ancient fragments of DNA that are found in very small amounts. All right, everyone, that's it for our video on concept number 4. Please leave for me at the end of the video any questions that you have that I can address when I return to class.